My name is Loredana Ricciardi. I am the managing editor of Asean Applied Sciences, and today I have the honor to have Dr. Ricciardi as our presenter. Thanks for accepting this invitation. Just before I give the word to her to introduce, of course, herself, her research, and her work, I would like just to give a very brief introduction to Journal of Asean Applied Sciences, if you didn't hear about it. So this journal is launched from January 2019. It is a multidisciplinary, interdisciplinary journal. And the aim to create it, to launch it, was to create a platform for the scientists who are working in different fields, including chemistry, material science, engineering, and earth and environmental science. The journal is open access, fully open access since January this year. And of course, if you had any more questions about the journal, submission in the journal, you can always, here you have my email address, you can contact me. And now back to our speaker, Dr. Ricciardi. If you can see, yes, you can see my slides. So I'm sure she will introduce herself better, but today her topic is about advances in cancer photo-theranostics. So she will talk about also the article that she published in our journal. Just I wanted to ask you, I can see everybody is actually muted and the cameras are off, but we sometimes have some connection problems. So please keep your cameras off. And at the end, you can just type your questions in the chat box and I can ask your questions and yeah, and we'll get your reply. Okay, thank you. So please just start. So I will stop sharing and you can share your slides.
Yes. Good morning, everyone, and thanks to Loredana for this opportunity and the kind introduction. Okay. Before going through the topic of today's talk, I would like to briefly introduce my research institute. The Institute of Nanotechnology of the National Research Council is a multidisciplinary center with more than 200 researchers coming from physics, chemistry, engineering, as well as biology and medicine, which develops fundamental and applied science in the fields of nanoscience and nanotechnology. In particular, the scientific activities of the research unit located in Calabria in close collaboration with the academic institution are focused on nanobiotechnology, material science, advanced device, photonics, and optoelectronics. In this framework, the topics that I personally deal with are study of the photophysical properties of chromophores by steady state and time-resolved spectroscopy, as well as the use of luminescence techniques to investigate biomolecular interactions. Then, as we will also see during this presentation, the other main topic of my research concerns the synthesis and characterization of nanoparticles, especially metal nanoparticles, for life science applications. In the paper recently published in Springer Nature Applied Science, we highlight advances in light-activated cancer theranostics by using hybrid combinations of two different classes of materials. Transition metal complexes, in particular based on ruthenium, iridium, and platinum, and the gold structure at the nanoscale. Was a common denominator is the ability to interact with the light radiation, catching the radiant energy and converting it into multiple other forms of energy very useful in diagnosis and treatment of cancer. But first, let's take a look at the properties of each class. So far, the application potential of transition metal complexes has been widely explored in the field of chemotherapy, photodynamic therapy, and bioimaging. Since the discovery of the antiproliferative properties of cisplatin in the 60s, platinum-based agents have been used in cancer therapy to treat several solid tumors, currently covering almost 50% of all the clinically used chemotherapy drugs. The mechanism of action underlying the therapeutic efficacy of the platinum compounds approved by Food and Drug Administration is based on their ability after intracellular activation to bind DNA of cancer cells, forming intrastrand cross-linked dots with the inhibition of their replication and activation of apoptotic processes. Due to their attractive photophysical properties, transition metal complexes have shown to be also excellent photosensitizer for photodynamic therapy application. They meet essential requirements for a photosensitizer, such as intense absorption in the visible region, high to photon absorption cross-section, and then a possible their activation by wavelengths within the biological window. Moreover, the presence of the heavy atom, the metal center, promotes spin-orbit coupling, leading to ultra-fast and efficient population of triplet excited states by intersystem crossing. Triplet states often very close to the singlet states of molecular oxygen, with consequent energy transfer processes and generation of singlet oxygen, a highly cytotoxic species, very reactive, which causes oxidative stress and lastly cell death. To date, the clinical application of transition metal complexes in photodynamic therapy is in its infancy, with the first ruthenium compound advanced to clinical trials in early 2017 for photodynamic treatment of bladder carcinoma, and currently undergoing phase 2 studies. Moreover, as I say, the high yield of intersystem crossing leads to an efficient population of triplet states that can drop to the ground level radiatively, giving rise to phosphorescence. Begin the electronic transition between the triplet excited states and the singlet ground states spin forbidden. Lifetimes are usually on the time scale of microseconds, making transition metal complexes long-lived hemisphere probes very useful for bioimaging, in particular in time-resolved photoluminescence techniques. As clearly shown in the figure, the long-lived luminescence signals allow to collect the light signal of probes during imaging in a time window beyond the short lifetimes of the endogenous fluorophores, minimizing interference and fluctuation in results. Furthermore, since the lifetime values of transition metal complexes are generally strongly dependent on the surrounding environment and oxygen content, by means of phosphorescence lifetime imaging microscopy, it's possible to map the localization of the molecules in live cancer cells, as shown in this example, where the square planar platinum complex used as a probe, upon 2 photon excitation, exhibits different emission lifetime if localized in the cytoplasm, 2.3 microseconds, or in the nucleus, 4.2 microseconds. Gold nanoparticles are one of the leading nanomaterials in cancer theranostics. They have proven to be excellent drug delivery systems and efficient nanosources of heat for photothermal therapy application, whereas the strong X-ray absorption cross-section and light absorption and scattering properties make them useful contrast agents for computed tomography, photoacoustic, and light scattering imaging. As nanocarriers, they have the ability to take advantage of the enhanced permeability and rotation effect, a phenomenon employed for passive targeting, resulting from the different vasculature system of tumor compared to the blood vessels of healthy tissues. In particular, the tumor region is characterized by leaky vessels with pores through the endothelial gaps between 100 nanometers to 2 micrometers, which promote the nanostructures crossing and tumor accumulation. Moreover, the poor lymphatic system limits their drainage, leading to an intratumoral rotation higher than in other tissues. On the other hand, active targeting can be performed functionalizing the nanoparticle surface with ligands for specific recognition by surface receptors, particularly overexpressed in target cells, promoting a selective addressing and bioavailability at tumor sites. Of course, the combination of passive and active targeting makes gold nanoparticles an ideal carrier for in vivo delivery of therapeutic and diagnostic agents. The most intriguing property of noble metal nanostructures is the localized surface plasma resonance, a unique property that enables nanoparticles with exceptional effects, such as electric field enhancements, huge light scattering and absorption at the resonant frequency, and the striking photothermal conversion capabilities. The plasma wavelengths are strongly affected by nanoparticle geometry, giving rise to colloidal solution of different color, just changing their size and shape. For gold nanoparticles, the plasma resonances fall within the visible near-infrared range. Then they are of particular interest for application in photothermal therapy. In fact, upon light exposure, they are able to generate local hyperthermia with consequent disruption of cellular membranes and protein denaturation. In this field, the great advantage of gold nanoparticles, in particular shape anisotropic particles, is such as gold nanorods and gold nanostars, is to exhibit plasma resonance peaks in the so-called biological windows, the spectral range where tissue become partially transparent due to a simultaneous reduction in absorption and scattering. The use of specific laser sources to excite the nanoparticles working in the biological windows allows deep tumor treatment due to a longer optical penetration. In the cutting-edge field of phototheranostics, the hybrid combination transition metal complexes gold nanoparticles could represent a synergistic merger of multiple functionalities, confining in a single nanostructure treatment strategy and imaging properties, providing the opportunity to exploit the cooperative interaction between them. Certainly, in the hybrid nanosystems, the properties of both components influence each other, often leading to an improvement of the individual properties. Moreover, the strength of the resulting nanoplatform is the ability to be triggered by light exposure with a spatial and temporal control of its activation for highly localized treatments. Now we will see some examples about the in vitro, in vivo application of transition metal complexes, gold nanoparticles, starting with an example of their use as imaging probes. The luminescent iridium complex bearing acylphenylpyridine has a cyclomethylated ligands and a bipyridine with long lengths has ancillary ligand was used to coat gold nanospheres with a size of 13, 25, and 100 nanometers, with the aim to develop novel nanoprobes for 2-photon lifetime imaging. The authors investigated in detail the 2-photon absorption cross-section of the hybrid nanostructure with the gold particle of 25 nanometer and observed the huge increase compared to the 2-photon absorption cross-section of the iridium complex molecule, attributing this enhancement to the proximity of the compound to the plasma nanostructure. Moreover, taking advantage of the iridium complex sensitivity to the cell environment with lifetime values ranging from 450 to 1000 nanoseconds, an intracellular localization map was obtained for all nanoparticle sites, highlighting the different internalization and presence of clusters. In the next example, transition metal complexes, gold nanoparticles, were used as imaging probes by means of a dual modal imaging, computed tomography and luminescence. In particular, an iridium complex and a gold nanoparticle with a size of 10 nanometers were encapsulated within poly-ioxol nanoparticles. Ioxol is a commonly used contrast agent, leading to composite nanoparticles with an average size of about 50 nanometers for in vitro and in vivo imaging. Gold nanoparticles were used to improve computed tomography and due to the proximity to the iridium complex as metal enhancement fluorescence effect agents, with a consequent improvement in phosphorescence imaging. The composite nanoparticles displayed higher contrast ability than ioxol and a clear outline of the tumor area after mice injection highlighted the good contrasting ability. In vivo, the metal enhancement fluorescence effect of gold nanoparticles increased the phosphorescence intensity in mice injected with the composite with respect to those treated with the transition metal complexes alone. Moreover, since the iridium complex luminescence is sensitive to oxygen content, the hypoxic tumor region showed a distinctly brighter red light than that of other tissues and organs. In the next example, we will see hybrid nanostructures used simultaneously for therapy, in particular photothermal therapy, and fluorescence imaging. The author reported an unexpected high photothermal conversion efficiency of gold nanospheres of 45 nanometers upon grafting with the 2-photon luminescence ruthenium complexes. They reported 3 samples of grafted nanoparticles differing in the distance between the ruthenium center and the gold surface. Then the photothermal properties of all the nanoparticles were investigated, showing clearly that the grafting enhanced the gold nanoparticles' photothermal efficiency compared to pure gold nanoparticles, in particular in the case of complex 2, in agreement with the fact that this compound had the highest 2-photon absorption cross-section and then high energy transfer to gold nanoparticles. The best dual functional nanoparticle grafted with the ruthenium-2 was used for real-time luminescence guide photothermal therapy in HeLa cells and to perform in vivo studies. In vivo experiments after injection of the selected nanoplatform followed by irradiation highlighted a complete tumor ablation, whereas the histological examination after treatment revealed a necrotic tumor tissues. Then we will see now a multimodal synergistic approach recently presented by our group, where a hybrid nanostructure was engineered for simultaneous photodynamic, photothermal therapy, and imaging. We have chosen as a transition metal complex and iridium complex bearing to cyclometallated phenylpyridine and an ethylene diamine has ancillary ligand. The complex is highly soluble in water and shows a bright emission in the green region of the electromagnetic spectrum with an emission quantum yield of 0.1 in presence of oxygen that increases up to a value close to unity in a durated solution, pointing out indirectly its remarkable ability to generate singlet oxygen. Moreover, the emission band overlaps with the plasmonic resonance of gold nanosphere, making possible an energy transfer process from the cyclometallated complex to gold nanoparticle, with consequent heat generation. Then, in order to obtain that and to have both photodynamic and photothermal effects, we prepared the gold core silica shell nanoparticles, embedding the iridium complex within the polysiloxane matrix. In vitro photocytotoxicity test of the hybrid nanoplatform performed on human glioblastoma cells were compared to those of the free iridium complex embedded within silica nanoparticles and gold silica nanoparticles without the photosensitizer, highlighting a high photocytotoxic ability of the hybrid nanoplatform already at very low doses and higher than that observed for the iridium complex alone, embedded within silica nanoparticles and of gold silica nanoparticles without transition metal complex, pointing out how the results come from the combination of photodynamic and photothermal effect. Then, in vivo study on glioblastoma xenograft mouse model after intratumoral injection of the hybrid nanosystem followed by irradiation at 365 nanometers displayed a complete tumor regression within 24 days post-treatment without recurrence. The luminescence properties of the transition metal complex allowed to track over time the in vivo distribution of the nanostructure, whereas the presence of gold made them efficient contrast agents for X-ray phase contrast tomography, employed to image the 3D tumor vascular network of ex vivo glioblastoma tissues in order to shed light on the mechanism underlying the tumor eradication. Another example of dual modal therapy, in particular photothermal therapy and chemotherapy and imaging, was reported by using an innovative nanostructure for intraoperative real-time Raman detection and chemophotothermal therapy of disseminated Hovarian microtumors. The nanostructure consists of a gold core and a gold shell separated by an internal sub-nanometer gap containing a Raman reporter, in particular benzoyl, and an external mesoporous silica layer with cisplatin molecules loaded into the pores. The developed nanosystem has photothermal therapy properties, fingerprint Raman signal from the molecules inside the nanogaps, strongly enhanced by the presence of the gold nanostructure, and chemotherapeutic effect due to the cisplatin molecules. In vitro studies revealed that after treatment, nanoparticles plus laser irradiation, almost 100% of tumor cells were killed. In vivo mice after nanoparticles injection underwent laparotomy surgery guided by Raman signals, easily detected from microtumors of various sizes disseminated in the abdominal cavity. And real-time photothermal therapy was performed on the positive Raman locations. Finally, in vivo therapeutic effect revealed a near complete tumor eliminations with a survival rate of 100% during the observation period. In the last example, the transition metal complexes gold nanoparticles allow a combined therapeutic approach, photothermal therapy and chemotherapy, and a three-modal imaging, photothermal, computed tomography, and photoacoustic. The reported iridium complex was loaded on gold nanostars, and in order to enhance the targeting ability towards triple negative breast cancer, the nanoplatform surface was functionalized with a peptide conjugate. The obtained nanostructure showed an efficient selectivity, a photothermal enhanced iridium complex release under near-infrared laser irradiation, and excellent photothermal, photoacoustic, computed tomography imaging abilities. In vivo studies on tumor-bearing mice after intratumoral injection, followed by laser irradiation, revealed an almost complete tumor regression. In conclusion, the combination of the appealing properties of both transition metal complexes and plasmonic gold nanostructures in a single platform provides the opportunity to use simultaneously various diagnosis and treatment modalities with final synergistic effects and enhanced therapeutic outcome. Effects triggered by light and then confined exclusively to the region exposed to irradiation, and where the nanoplatform have been passively and actively targeted, leading to highly selective therapeutic approaches. Thank you very much. Thank you again the organizer, and of course, thank you very much for the attention of all the participants.
Thanks a lot, Loredana, for very nice presentation. So now this session is open for questions. Please just type your question in chat box so I can read it. But I will just start with one question, because all of these nanostructures are using transition metals, but also it means noble metals, platinum and gold. So they are, of course, costly and expensive. I'm just asking myself, is it possible to replace these noble metals with cheaper metals, like other transition metals, which have probably similar characteristics, or not?
Yes, we hope to do this, but of course the properties are strongly dependent of the metal that we use in the complex. Of course, ruthenium, iridium, platinum are the complexes that exhibit a better performance, of course, in photodynamic therapy. In particular, ruthenium and iridium are the most used for their important properties.
Okay, yeah, I see. Usually that's the problem in any field. So you cannot replace these noble metals easily with other transition metals.
Exactly.
I have a question here. Are these nanostructures used at the moment for real patients, in the hospital just for treatments, or this is mostly at the moment just in the research?
just in the research. There are some nanoplatforms in clinical trials, but to date, there isn't a Food and Drug Administration and approbation for using clinical up to date.
Okay, so still
but
there's
we hope, yes, I think that it's a very innovative combination that, of course, the advantages are a lot, and we have just to wait, and I think few years just to have the first clinical approved, I think, combination.
Okay.
Just for the great potential.
Yeah, I see. Another question, which other working groups in the world just are working on these nanostructures, like that you have probably collaboration or they are working independently? Do you know about this?
We are a lot of groups in this field because
Okay.
yes, in Italy, but of course also in the USA and in France for my collaboration. That's just my personal point of view. It's a field very, very interesting. So I think a lot of groups are in this contest in this moment.
I see. I see. So please just, if any questions, you can type in the chat box. I still don't see. Let me check. Not yet. So I don't know if you want to add anything which you didn't present in the just during your presentation, any additional points also, because I don't see any questions. These were the only ones. So, okay. So if everything is clear apparently and nobody has any question, but of course, yeah, everybody, if there was any question, people can contact you probably and ask. So thanks again for the presentation and accepting our invitation, and thanks all the participants for listening. I mean, this video will be also broadcast online. I will share the link with you and the participants later, so it's possible to again just share it with others. Have a nice week and a day, and hope to see you again in another conference.
With a great pleasure.
Thank
we hope
you.
very, very, very soon. Thank you.
Thank you. Bye.
Bye.